Hello, everyone. It's my great honor to present to you the research findings published by the research group led by Professor Su-Chun Lan from Northwestern Polytechnical University in the journal iMeta. The title of the study is Hypertriglyceridemia Modulated Gut Microbiota Promotes Lysophosphatidylcholine Generation to Aggravate Acute Pancreatitis in a TLR4-Dependent Manner. First, let me briefly introduce the research background. Acute pancreatitis mainly results from abnormal activation of pancreatic enzymes, causing self-digestion of pancreatic tissue. Its causes include cholelithiasis, alcohol, etc. In recent years, with social and economic development, hypertriglyceridemia has become the third leading cause of acute pancreatitis. Patients with hypertriglyceridemic acute pancreatitis are more likely to develop severe cases, suffer from persistent multiple organ failure, and have a higher mortality rate, but the mechanism of how hypertriglyceridemia worsens acute pancreatitis is unclear. So, this study aims to explore the role and mechanism of gut microbiota-host interaction in this process, starting from the gut microbiota. In this study, we found that in hypertriglyceridemia mice, gut microbiota changes led to a rise in endotoxin levels. Hypertriglyceridemia worsened intestinal barrier function in acute pancreatitis mice, increasing bacterial translocation risk. Also, the hypertriglyceridemia-regulated gut microbiota promoted lysophosphatidylcholine production in a TLR4-dependent way, exacerbating acute pancreatitis. Our main findings: we created a hypertriglyceridemic acute pancreatitis mouse model. Compared with acute pancreatitis, these mice had worse pancreatic injury and inflammation, showing hypertriglyceridemia worsens it. The 16S rDNA sequencing results showed that HTG could lead to gut microbiota dysbiosis in mice and an increase in endotoxin content. To explore the gut microbiota's role in hypertriglyceridemia exacerbated acute pancreatitis, we depleted relevant gut microbiota with antibiotics and did fecal microbiota transplants. Depletion eased inflammation and pancreatic injury, while transplantation from hypertriglyceridemia mice worsened them. Gut microbiota interacts closely with the host. To explore its impact on mice under hypertriglyceridemia, we used non-targeted metabolomics to analyze serum metabolites. After gut microbiota depletion, 17 metabolites rose and 64 fell. KEGG analysis showed stronger glycerophospholipid metabolism and a significant increase in lysophosphatidylcholine in serum and pancreas. Phospholipase A2 limits lysophosphatidylcholine production. Prior research shows TLR4 activation upregulates PLA2, with endotoxin activating TLR4. To explore how hypertriglyceridemia-regulated gut microbiota controls lysophosphatidylcholine production, we isolated primary mouse pancreatic acinar cells and made an in vitro model with LPS exposure. After treating with TLR4 inhibitor TAK-242, TLR4 signaling, PLA2 content, PLA2 gene transcription, and lysophosphatidylcholine levels all dropped. Meanwhile, the same occurred in TLR4 knockout mice. After knockout, in acute pancreatitis mice with hypertriglyceridemia-regulated gut microbiota, TLR4 signaling, PLA2 content, PLA2 gene expression, and lysophosphatidylcholine levels all dropped, relieving pancreatic injury and inflammation. To further explore lysophosphatidylcholine's effect on acute pancreatitis, we supplemented it externally to affected mice. Results showed this worsened pancreatic injury and inflammation. In this study, we found that hypertriglyceridemia exacerbates pancreatic injury and inflammatory responses in acute pancreatitis mice in a gut microbiota-dependent manner. Hypertriglyceridemia can cause disorders in the composition and function of the gut microbiota in mice, leading to an increase in endotoxin content. The gut microbiota regulated by hypertriglyceridemia promotes glycerophospholipid metabolism and lyso-PC production in a TLR4-dependent manner, thus exacerbating the severity of acute pancreatitis. That's the main content of this study. If you have any questions, feel free to contact the